Hello everyone, this is Nevetha, Assistant Professor, Department of Biotechnology, Kaveri College for Women Autonomous. I am going to brief on CALLUS culture. A brief history of CALLUS culture. R.J. Gauthiret, he is actually a French scientist and he was the first successful person in promoting the development of callus tissue that has been excised from cambial tissue of Salic capreae. That is actually a woody plant. And he was also able to promote the growth of callus tissue using simple nutrient media that is supplemented with just 3 vitamins. There are also other fellows who contributed to this CALLUS culture: Norbert Koch, Van Overbeck, Kaplan, Skoog and Miller. What does a callus tissue means? Callus means an unorganized proliferative mass of cells and that has been produced from an isolated plant cells or tissues or organ. That too when it is grown aseptically in an artificial nutrient media, either in glass vials or in controlled environmental condition. Principle of callus culture. It has 3 main principles. like aseptic preparation of plant materials, and selection of suitable nutrient media. That this nutrient media should be supplemented with equal ratio of auxins and cytokinins. And we have to incubate the culture under controlled physical conditions. So the next is procedure, and the first step is surface sterilization. So before attempting to initiate a callus culture, it is very mandatory that we have to surface sterilize the plant part whichever to be cultured. That is why, because plant parts carries number of surface bound microbes like bacteria, fungi and etc. So the excised plant part should be cut into smaller pieces, and then it should be rinsed in under tap water, and then subject the explant into an liquid detergent like T-Pol, Tween 20, etc. Then the explant are surface sterilized by most commonly used chemicals like mercury chloride or sodium hypochlorite. That is, we have to maintain it in for limited period of time. Generally 10 to 15 minutes is enough. And after surface sterilization, the explants are need to be rinsed repeatedly with autoclaved distilled water. The surface sterilized plant material is cut into smaller pieces like smaller segments, and the size of the explant is very critical factor for the induction of callus tissue. So this is the image. The explant should be finally transferred aseptically into a suitable nutrient media, either solid or semi-solid nutrient media, and we have to wash it. The media, whatever we use, need to be autoclaved at least for 15 minutes. We are drying it. So the success of the callus tissue involves in the composition of the media. Explant is mainly growth hormones is required in the nutrient media for the healthy callus growth, and you have to maintain the auxin and cytokinin level. And it has to be incubated under a proper physical conditions like temperature, light, humidity have to be taken care. In some plants, it requires darker condition for callus to grow, and for some photo period is somewhat different. 16 hours light and 8 hours darkness is need for the callus initiation. Once the callus is grown, well-established callus can be transferred to another fresh nutrient media for the continuous growth purpose. In this way, the callus culture is continuously maintained by series of subculture. Next is significance of callus culture. The whole plant can be regenerated from a small portion of callus and even in larger numbers. Callus is also a good source for genetic and karyotyping variability, and suspension culture can be initiated from the callus culture. And finally, callus culture is very important and useful method for obtaining commercially important secondary metabolites. Thank you for watching this video.